My name is Dalia Perez. I'm a postdoctoral fellow here at the Koch Institute working in the Lees lab. In the Lees lab, we study biological pathways, often mutated in tumors, and we're interested not only in how these pathways function in normal biology, but also how mutations in these pathways can contribute to cancer. So we set out to study uveal melanoma, which is a particular subtype of melanoma found in the eye. So skin and uveal melanoma initiate in the same cell type, namely the melanocyte, but the mutations contributing to uveal melanoma are genetically distinct from those that contribute to cutaneous melanoma. Because of this, treatments for uveal melanoma that are commonly extrapolated from skin melanoma patients are not efficacious. And prior to this work, it was reported that over 80% of uveal melanomas had initiating mutations in genes called GNA11 or GNAQ. So we developed this model by inserting the human cancer-causing mutations into the fish genome in such a way that only the melanin-producing cells of the fish would express this mutation. So we've known for about 100 years that fish can get cancer. Actually, the first report in the 1920s was a melanoma case. And importantly for studying melanoma, the zebrafish also have an equivalent of a melanin-producing cell called a melanophore. So this is actually one of my first attempts at imaging the melanocytes in the fish. And you can really clearly see the melanocytes, which are the kind of heterogeneous, brown, kind of dendritic and circular cells here in the picture. And you can see them alongside another yellow pigment-producing cell in the fish called the xanthophore. And it's the interaction and the signaling between the melanophores and the xanthophores in the fish that gives the zebrafish that beautiful stripe, interstripe pattern in the wild type fish. So importantly, a lot of the signaling pathways normally used by melanocytes in the wild type fish to set up the kinds of stripe patterns that I'm talking about are pathways that are commonly hijacked by cancer. And so it's these kinds of pathways that I'm interrogating by designing this set of experiments. So in this way, I could ask, what is the effect of having the GNAQ or GNA11 mutation on melanocyte biology in the zebrafish? And when we inserted the mutated GNAQ or GNA11 into the fish, I saw this incredible pigmentation change in the skin of the fish. So once I saw these pigmentation changes at the organismal level in the fish, the next thing that I wanted to do was to see what was happening to the melanocytes at the cellular level. So from this baseline, I was able to design experiments to look at changes in melanocyte morphology, motility, signaling, and proliferation in the melanocytes upon introduction of this mutation that initiates uveal melanoma development. We need insights into the mechanisms of tumor signaling downstream of these mutations to finally get to some of the more targeted therapeutic strategies that clearly these patients need.